Okay, so let's try this one more time. Thank you all very much for coming today to Bits in Bio Philadelphia presents Breathe in Bio. It has been one of our more impetus and initiatives to get us to grow as a chapter this year. We have a lot to discuss and a lot to go through. So we're excited to talk to our speakers and to allow them to talk more about their work, including some of the exciting areas of biotech that they are going to talk about. Excuse me for one moment. Thank you. Sorry, I just had to admit people in, and it's kind of hard when Zoom decides to hide some of these controls under a mouse. So anyway, to talk about Bits and Bio for a second. So Bits and Bio's mission is a community-building software for science. A lot of it focused on not only bits and bios and biotech, but also tech bio. The mission is to accelerate development of bioeconomy, educating and empowering the next generation of biodevelopers, whether it is from a lens of a startup to being a great product designer, data scientist, wet lab, dry lab, whichever way you think about it, empowering these people to adopt computational data science, computer-based methods inside the business. We started in 2023, the major organization with over 2,000 members, and it has exploded. It has been more than just what is a Slack channel, which is what we use. It has also been a community for underserved groups of technologists, biologists working in the industry. And so there is a lot to be done, and we're really excited to do that. Um, just to kind of put it out there, it is Bits and Bio is a nonprofit in the biotech tech bio space. Again, the community has doubled. We have meetups, whether it is in the New York area, San Francisco, Philadelphia, all the other areas. We're really excited to continue this growth as an organization. And the organization has things like Q&A, blog posts, lightning talks. They publish a survey that will be coming out soon, which is the state of tech bio. Particularly for the Philadelphia chapter, we have a lot of things that we're going to try to do throughout the years. So if you're in the Philadelphia area, we have a happy hour. We're going to start it again in February at 5 p.m. I have the Luma link that we're going to post down in the chat and just start get that disseminated. We're going to have a lot of events. And if you want to get involved in the chapter, we want you to just do us a favor and just message me or contact me. It'll make things a lot better, and we'll be excited to get you involved in the chapter. So give me one moment. Sorry, we're there's a little bit of a tech difficulty with one of our presenters. They're trying to join, and I don't know why, but Zoom is not being simpatico with that. But if you just give us 1 second, and then we will get to our speakers. So if you don't mind, I'm gonna pull up this slide once again. So today we have two great speakers in Sesan and Meraj. Sesan is coming from iLABs, and Meraj is coming from DigiBiomics, both of them being co-founders and CEOs. Rather than just give an introduction, we're gonna go from what I consider left to right, and let Sesan introduce himself, talk more about what he does, and then we'll go over to Mirage, and then I'll field a couple of questions. We'll have the audience get into questions, and we'll go from there. Let's get it started with Sesan.
Yeah, thank you very much, Ade, for the kind introduction. It's a pleasure meeting everyone, and my name is Sesan Ajina. I am the co-founder and CEO of iLABs, and we use disease modeling and live cell imaging to build a cell analysis platform that helps biologists build a lot faster than they would otherwise. Um, I come from like an unorthodox background to biology. Everything I know about cells and biology is very hands-on, and I feel like I have a very interesting job that I have to observe cells and see how they behave, and that's very exciting. Okay, so should I go ahead with a presentation?
Yes, and then we'll go to
All right,
Mirage next.
awesome. So, the topic of my presentation today is an introduction to web-based cell analysis, and why is this important? Sorry, can you do the next slide, please? So with microscopy, it's a very, very uh, complex or straightforward concept, depending on where you're coming from. And it plays a very critical role when it comes to like biological research. It typically can be broken down into two fundamental components, which is light microscopy and electron microscopy. The key difference there is light microscopy uses light to illuminate samples, and electron microscopy uses electron. There's a trade-off there. With electron microscopy, you get a lot more detail. Sorry, first, not yet. Sorry. With electron microscopy, you get a lot more detail, meaning you get it blown up and a lot more resolution than light microscopy, but there's a trade-off there. So if you have electron microscopy, you're going to lose a lot of speed. But with light microscopy, which is what most of you guys are probably familiar with, you have a lot of speed and you can analyze a lot of samples subsequently. Sorry, next slide, please. Um, so live cell imaging. Um, there is a trend with microscopy for researchers moving to live cell imaging and multimodal imaging. And the reason for that is just very straightforward. You can observe organism in their natural environment. So that way, rather than trying to postulate what the mechanism or disease mechanism is, you could literally just observe and try to make your inference from actual cellular behaviors. It's a very powerful technique for non-invasive analysis of cells, and it reveals biological processes and hidden mechanisms. You could do a lot of like single cell analysis, looking at how each cell behaves individually in response to to some form of therapy, and that way you could try to identify drug targets from just seeing how cells behave over time. Next slide, please. Uh, so the powerful combination here is by combining disease modeling and AI. So the idea here is to develop a platform that analyzes cellular behavior in the context of a disease progression. So that way you could sort of like do a much more targeted approach to whatever disease you are trying to cure. Um, by combining live cell imaging and disease modeling, you are sort of like power charging the approach for trying to develop a um, disease treatment from a cellular level. Because you have to remember, cells are the foundational units of life. So if you could understand how diseases emanate on a cellular level and you could design a cure for that, you have a very good shot at actually running a cure that actually passes a lot of the tests. The screenshot you see right here is actually from our platform and for analyzing a two-dimensional time series data. And you could see there is you are extracting cellular features like the information. And this information is critical because it's in response to a therapy or treatment. So that way you could see the changes in the morphology or changes in the dynamics is like the speed and the distance that each of the cells cover, which is in response to some sort of therapy or treatment that's been applied to the cell culture. So, next slide, please. Use cases and demos. There are lots of potential use cases in this context, but at the moment we are focusing on human cancer cell research. And we're doing that by helping researchers quickly identify drug targets. And we do that by developing disease models for different types of diseases. And we do this by taking cells, cancer cells to be very specific, and we develop a model that could actually identify these cells and track, measure, and extract features of the cells throughout the course of your environment. This is relates to this community in the terms that we're working with, like small lung cancer cells, the A549 cells, and they have very, very interesting morphology. So a lot of the research that scientists are doing right now is testing different compounds and using the morphology and viability properties to see what drugs or compounds they could actually pass down the pipeline. Because these properties are very, very key factors to see how the cells are actually responding, either positively or negatively, depending on the context. And the second use case is the basic understanding of disease mechanism. So the fact that you have the opportunity to observe cells in a dynamic environment, dynamic natural environments to be precise, you could just see exactly how they react to different stimulants. And this gives you the opportunity to develop or design disease progression algorithms that you could actually use as a form of analysis to see how each of your experiments are progressing. Uh, an example here is where we use an application for neurons, N1E, which is a cell line, a neuronal cell line, and it was for a electro stimulation therapy. So this is like a condition where you actually do like little electrical shocks to neurons, and you try to see how that actually affects neurons. And this has been um, proposed as a potential treatment for, like, chronic back pains and some um, really deep spinal conditions. And the initial conclusion was that this was going to be helpful. And the key factor that was observed was how quickly the cells moved, which is a very um, directional movement. So there is a dimension that we measure called directness. So directness is how the cells are moving in a particular direction in response to that therapy. So if the cell is moving in a straight line, it means that that's a cell that knows exactly what it's doing and it's responding to the therapy, as opposed to like a cell that's like moving in some sort of like a, you know, non-determinate way, like a zigzag kind of movement, that is considered random. So you would analyze how the cells are moving, whether they're direct movements or a random movement. And based on that classification, you could determine how the cells are responding to that therapy. So these are some of like the um, use cases that we have currently right now. And my hope is that we will get some of you guys who are hopefully working on some cell culture or cellular research for lungs or different type of lung cells. We are onboarding users right now. We would love for you to test the platform if you have some data. And I look forward to all of your questions and feedback to my presentation. Thank you very much.